Hello students, this is your Prasanna ma'am from NEET World. Here we are going to discuss the topic fungi from the chapter biological classification. After keenly looking into all the previous year questions of NEET, we have sorted and listed out the topmost important gunshot topics for your NEET exam, okay, of which the first topic we are going to discuss, that is fungi, okay. If you look into the general characteristics of fungi, who introduced the kingdom fungi? Firstly, tell me who introduced the kingdom fungi. It is R.H. Whitaker. Who he? R.H. Whitaker in the 5 kingdom system of classification. In which kingdom beta? In 5 kingdom system of classification. He has introduced the kingdom fungi, okay. What are the general characteristics of fungi? Fungi are eukaryotic, multicellular, thalloid. What does the term thalloid means? Thalloid means there is no organ or organ system level of organization They are all achlorophyllous and purely heterotrophic organisms. And if you see, there is great diversity in morphology and habitat in the kingdom fungi. There is a great diversity shown in the morphology and habitat of fungi. Morphology, if you see, you can see from unicellular yeast, that is Saccharomyces cerevisiae, to complex edible mushrooms and toadstools, okay. If the question is asked like most metabolic diversity is shown in which organism or which kingdom, then your answer should be Monera. Metabolic diversity is shown by the kingdom Monera. Diversity in morphology and habitat is shown in the kingdom fungi. Next, if you look into the habitat, they are cosmopolitan in distribution, but they mostly occur in warm and humid places. Next if you see hyphae, what does fungi made up of? Fungi are made up of hyphae, okay. Fungi are made up of hyphae. Hyphae are nothing but small thread-like long strands. They are slender and thread-like. This network of hyphae, you will call it as what mycelium. Network of hyphae is mycelium. Few mycelium are cenocytic, means they are aseptate. The hyphal strands are not divided by septa, they are cenocytic. Cenocytic means what? Multinucleate condition. Multinucleate condition of hyphae. Multinucleate or cenocytic condition. Cenocytic condition. It is seen in aseptate mycelium. It is seen in what type of mycelium? Aseptate mycelium, where you will see cenocytic hyphae and continuous tubes which are not divided by any septa. Such type of aseptate mycelium you will observe in Phycomycetes. Such type of aseptate mycelium, where do you observe it? In Phycomycetes. And few mycelium, they are septate. The hyphae are divided with cross walls or septa, okay. They are divided by cross walls or the septa, and you will see each septa is having one nucleus. And hyphal body, if you see, it is haploid body, okay. Fungal body is what Fungal body is haploid, which means it has only half a set of chromosomes, okay. In septate mycelium, you have different types of septa. If you see ascomycetes consists of pore-like septa. What does it mean? The hyphae are divided by pores like this. Hyphae are divided by what beta? Pores. If you see basidiomycetes, dolypore septa is present. What is dolypore septa? If you take a hyphae, small sac-like structures that divide the septa. This is called as what? Dolypore septa. Pore-like septa you will see in ascomycetes, dolypore septa you will see in deuteromycetes. in Basidiomycetes. Doliprosepta septa is seen in what? Basidiomycetes. Deuteromycetes are also have septate mycelium, but there is no specific septa. Okay. Yes, under septate mycelium, what are the classes we have? Ascomycetes, Basidiomycetes and Deuteromycetes. Ascomycetes has spore-like septa, Basidiomycetes, Doliprosepta septa, Deuteromycetes are septate mycelium. Next, the cell wall of fungi. What is the cell wall of fungi composed of? Chitin, which is called as fungal cellulose. Chitin and polysaccharides, this is called as what ma? Fungal cellulose. Fungal cell wall is made up of chitin and polysaccharide, it is called as fungal cellulose. Nutrition is purely heterotrophic. Nutrition is what beta? Purely heterotrophic, of which we have three types of heterotrophic nutrition. One is saprophytic, other is parasitic and then symbiotic type of nutrition. If you see saprophytic, how does fungi conduct saprophytic mode of nutrition? If you take hyphal strands or mycelium, they will release some enzymes. What do they release? Enzymes. These enzymes will digest the complex organic matter which is present over here. They will digest the complex organic matter into simple inorganic matter, into simple inorganic matter or substance. This is consumed by the fungi. The simple inorganic substance is consumed by what? Fungi. What does saprophytic mode of nutrition say? Fungi or the hyphae or mycelium, it will release out the enzymes. Those enzymes will break down the complex organic materials into the simple inorganic substances. These simple inorganic substances are consumed by what? Fungi. Okay, this is saprophytic mode of nutrition where they rely on dead and decaying substances. Where they rely on what ma? Dead and decaying material or matter, dead and decaying matter. Okay. Parasitic, they are relying upon the living host. They are relying upon, the nutrition is relayed upon living host or living organisms. It can be either plants or animals. This is pathogenic, wherein we call it as what ma? Fungal infections. We will call it as what beta? Fungal infections, both in plants and animals. Okay. Symbiotic mode of nutrition happens when they are associated with algae. Algae associated with fungi is called as lichen. Algae associated with fungi called as what? Lichens. These are pioneers in the succession. Okay, the lichens act as what ma? Pioneers of succession, plant succession. Fine. And lichens, they can indicate, they are good indicators of pollution as well. Lichens are what? Symbiotic association of fungi and plants. If you look into mycorrhiza, fungi associated with roots of higher plants like pinus. Okay, fungi associated with roots of higher plants like pinus. This association of fungi with roots of higher plants will increase the surface area. What does it do? It will increase the surface area. Thereby, the absorption of water and minerals is also increased. What happens? Thereby, the absorption of water and minerals is increased. Okay. These are the symbiotic associations of fungi. After discussing the nutrition of fungi, let us enter into the reproduction which is happening in the fungi, which is most important, multiple times asked in the exam. The most questions appeared from this segment of fungi itself. Pay keen attention and listen, students. We have three types of reproduction, fungal reproduction, that is vegetative, asexual and sexual. What are they beta? Vegetative, asexual and sexual. Okay. Vegetative mode of reproduction is conducted either by fragmentation, fission and budding. Okay. When it comes to asexual reproduction, happens through the release of asexual spores. Okay. Asexual spores can be conidia, sporangiospores and zoospores. Of these three, conidia are the only exogenous spores. What are they ma? Exogenous. I will tell you what does exogenous and endogenous means. Okay. As of now, do remember, among these three asexual spores, conidia are exogenous. Add on one more asexual spores, those are aplanospores. Those are aplanospores. Okay. And among all these four, only zoospores are motile. Only zoospores are motile. Else all are what ma? Non-motile. So, do remember, from asexual spores, it is conidia which is the only exogenous spore, and it is zoospore that is the only motile spore. When we consider the sexual spores include oospores, ascospores and basidiospores. Oospores are again non-motile, endogenous. You will observe these in Phycomycetes. Where do you see this? In the class Phycomycetes. Ascospores you will observe in Ascomycetes fungi. Ascomycetes. These are also endogenous only. In these basidiospores here are the only exogenous spores. Only exogenous spores. You observe these spores in which class of fungi beta? Basidiomycetes. Yes, if you keenly observe the types of reproduction in detail, firstly it is vegetative reproduction. We learned, right, vegetative reproduction includes fragmentation, budding and fission. What does the term fragmentation means? If you look into a fungal mycelium, older fungal mycelium, you will call it as older hyphae or mycelium will break into fragments. It will break into fragments like this. Okay, it will break into fragments. Each fragment falling onto the substrata, will develop into a new hyphae. This will develop into what beta? This will develop into a new hyphae or new mycelium. This will develop into what ma? New hyphae or new mycelium. Next type of vegetative reproduction is budding. On a matured mycelium or on a matured bud, we will call that matured mycelium or that sac as mother cell, over which buds are formed. Over which what are formed beta? Buds are formed. These buds again they will disintegrate from the mother cell. They will fall on the suitable substrata. Under favorable conditions, they will develop into a new mycelium. Under favorable conditions, they will develop into what beta? A new mycelium. Clear? Fine. Next, third type of vegetative reproduction is fission, wherein a matured fungi or a matured fungal cell divides into two. Just like that, it divides into two daughter cells. This is called as amitosis also. This is called as what, Nana? Amitosis also, as there is no involvement of spindle fibers or spindle apparatus. Next, if you see asexual and sexual reproduction of fungi in general. In asexual reproduction and sexual reproduction, I said you right, different types of spores. If it is asexual reproduction, we have conidia, sporangiospores, zoospores and aplanospores. This mycelium will develop fruiting bodies over it. What does it develop? fruiting bodies over it. This fruiting bodies will release spores. This fruiting bodies will release what, Nana? They will release spores. If it is sporangiospores, in this sac-like structure called as sporangium, sporangiospores are released. Mycelium is haploid. I already told you fungal body is haploid. This sporangium is also haploid. Sporangiospores are also haploid. These spores, when falling on the substrata, they will germinate again into the mycelium. If you take exogenous spores, the only exogenous asexual spore is what? Conidia. If you take a mycelium, this conidia are formed like this, okay, on the conidiophores. On what, beta? Conidiophores. Conidiophores. Conidia are released exogenously. How they are released? Exogenously. They are not formed inside any sac-like structure like sporangiospore. They are liberated outside, right? So, these spores, when falling onto a suitable substrata under favorable conditions, they will germinate into a new mycelium. This is about the asexual reproduction. If you look into the sexual reproduction, this fungal body gives rise to again fruiting bodies like the sac-like structures, okay, fruiting bodies. These fruiting bodies, if they are ascocarps, again in the sac-like structures, they will fuse, they will fuse and they will form ascus. What do they form? Ascus, which is diploid, okay. From this ascus, zygote is formed inside the ascus, and in fungi zygotic meiosis takes place. Zygote is diploid, right, and this undergoes meiosis or reductional division, releasing 4 ascospores. This ascospores falling onto a suitable substrata, they will develop into a new mycelium. If you see here, ascus or heterokaryotic stage. If you look into the sexual reproduction keenly, there are 3 substages, beta. One is plasmogamy, second is karyogamy, and third is meiosis, okay. Plasmogamy is first protoplasmic fusion, okay, protoplasmic fusion between 2 fruiting bodies or 2 compatible mating types. After the protoplasmic fusion, the nucleus which is present in them is fused and forms at the initially in ascomycetes and basidiomycetes especially, initially dikaryon stage is formed. Dikaryon stage is formed. This forms a zygote after karyogamy. After nuclear fusion, what is formed? Zygote is formed. This zygote will release out what? Spores. What are they? Sexual spores like ascospores, basidiospores, oospores and zygospores as well. They, after falling onto the suitable substrata, develop into a new mycelium. In sexual reproduction, meiosis or reductional division is taking place. In asexual reproduction, only mitosis is taking place. In sexual, meiosis or reductional division is taking place. This is our brief understanding of asexual and sexual reproduction in fungi. Let us understand more detailedly for each class of fungi. Next, if you see, for asexual and sexual reproduction, we have different types of spores. Of course, we have discussed in detail. Zoospores are motile and biflagellate. Zoospores are what, beta? Motile and biflagellate. Conidia, motile and biflagellate. Conidia, they are non-motile and exogenous. They are released or formed at the tips of conidiophores like this. They are formed at where? At the tips of conidiophores like this. Okay. Sporangiospores, they are formed inside the sac called as sporangium. That is why they are thin walled, non-motile and endogenous. If you see oospores, they are non-motile and endogenous. Oospores are what beta? Non-motile and endogenous. Ascospores, non-motile, endogenous. They are formed in sac-like structure, that is ascocarp. Inside what they are formed? They are formed inside ascocarp. Next, basidiospores, they are non-motile, exogenous, they are formed outside the basidiocarp like this. Where they are formed? Outside the basidiocarp. Okay, done. Now, if you want, you can take a screenshot of this. It includes all the types of spores. Here under asexual, you can also add aplanospores beta. Here you can also add aplanospores. These are non-motile, endogenous again. Here if you want, you can also add zygospores. Okay, zygospores. Fine, non-motile, endogenous. Now, next if you see different classes of fungi. How different classes of fungi are formed? Based on the morphology of mycelium, mode of spore formation and fruiting bodies. Based on what criteria? Morphology of mycelium. At beginning general characteristics we have learnt, right? Morphology, they exhibit great diversity in morphology and habitat. Based on that morphology and based on spore formation, just now we have learnt how many different types of spores are there for asexual and sexual, and fruiting bodies, how fruiting bodies are formed, few by fusion, that is sexual reproduction, few without fusion by mitosis, that is under asexual reproduction. Keeping all these criteria in mind, fungi are classified into 4 classes. One is Phycomycetes, then Ascomycetes, third is Basidiomycetes, 4 are Deuteromycetes. Again based on the spore formation under sexual reproduction, Phycomycetes are classified into Oomycetes and Zygomycetes. Oospores are formed in Oomycetes, Zygospores are formed in Zygomycetes. Next, if you look into Phycomycetes in detail, Phycomycetes is also called as what beta? Algal fungi. Phycomycetes is also called as algal fungi or primitive fungi. It is also called as primitive fungi. This primitive fungi is found mostly in aquatic habitats and on decaying woods and as obligate parasites. This mycelium is aseptate and cenocytic. I already said, there are continuous tubes, continuous tube-like structures without any septa, continuous multinucleated tube-like structures. What are they beta? Continuous multinucleated tube-like tubes, you can say multinucleated tubes, or cenocytic condition. What are they? Cenocytic condition. So here under asexual reproduction, what are types of spores formed in Phycomycetes? Zoospores, which are motile. Zoospores are what? Motile. Aplanospores are non-motile. Both of them are endogenous only. Next, if you see sexual reproduction, just a minute, sorry, yeah, if you see sexual reproduction, it is of course by the fusion, right? Fusion of 2 gametes, 2 mycelia of compatible types. For example, let us take 2 mycelia of compatible types. Let us take this as plus strain, this as minus strain, okay, plus strain and minus strain. Both these plus strain and minus strain will release the gametes and these gametes will fuse. These gametes can be of similar in structure and physiology, that is called as isogamy. They can be dissimilar either in structure or in physiology, anisogamy, or male motile gamete, for example male zoospore like motile gamete and female male motile fuses with non-motile large female, non-motile large female. Motile male gamete fuses with non-motile large female, that is called as oogamy. So, fusion can happen in any of these three types. One is isogamy, where male gamete and female gamete are structurally and physiologically similar, or anisogamy, male and female gamete may be structurally or physiologically different. Oogamy, motile male gamete fuses with large non-motile female gamete. After this fusion, any of the types of fusion, the products formed under oomycetes, it is oospore. Under oomycetes, it is oospore. Under zygomycetes, it is zygospore. So, both oospore and zygospore are what beta? Diploid. Oospore and zygospore are diploid, and these are the site of meiosis. Site of what ma? Meiosis. So, when we are discussing about reproduction in brief, we have learnt that there are three stages of sexual reproduction. One is plasmogamy, second is karyogamy, and third is site of meiosis, or third is meiosis. Here dikaryon stage is not present in Phycomycetes. There is no dikaryon stage. There is no dikaryon stage. It is because immediately after protoplasmic fusion, karyogamy takes place. This diploid cells will form, this diploid zygospore and oospore are formed immediately after plasmogamy, and they are the sites of meiosis. Immediately they undergo meiosis and they will release the spores, okay, and they will release what? Spores. This is how sexual reproduction in Phycomycetes takes place. Next, if we see, if we look into the examples of Phycomycetes. To remember the examples of Phycomycetes easily, I will give you a small mnemonic RAM, R-A-M, for Phycomycetes, wherein R for rhizopus, which is bread mould, A albugo, white spots on mustard leaves, and M mucor. Okay, if you remember this one word, you can remember the examples of Phycomycetes. Next fungi is what? Next class of fungi is Ascomycetes, which is also called as sac fungi. It is also called as what? Sac fungi. It can, in this high morphological diversity you will see in this class of fungi, like from unicellular yeast Saccharomyces to multicellular like multicellular Neurospora, Penicillium, Claviceps, everything you will see under Ascomycetes. These are saprophytic, mostly decomposers, parasitic and coprophilous. What is the term coprophilous means? Those fungi which feed on dung or which lay on dung. They rely on what? Dung. Okay, which live or the habitat is on dung, then so it is called as coprophilous. Mycelium is septate, and you will see pore like septa. Mycelium is septate, and you will see what septa? Pore like septa, like this, pore like septa. Next, if you see asexual reproduction in fungi. Asexual reproduction in fungi, I already told you, fungi in Ascomycetes conducts asexual reproduction by the release of what? Conidia, by release of conidia. This conidia is released through conidiophores. If this is the hyphae, they produce conidia exogenously by forming fruiting bodies, which is called as conidiophore. What is that fruiting body? Conidiophore. Conidiophore will release conidia or form conidia outside, and they will liberate conidia out. When these conidia are released exogenously or liberated exogenously, they fall on the suitable substrata and they will germinate into mycelium. They will germinate into what? Mycelium. This is the microscopic view of how conidiophores are formed. See how nicely These are conidiophores, and this is how conidia is releasing all the conidia exogenously. Okay, clear? Next, the sexual reproduction of Ascomycetes includes, it is very interesting, look into it clearly. The female hyphae which bears the female gamete is ascogonium. It is what ma? Ascogonium. See here, this is the mycelia which is bearing ascogonium, that is female gamete. Okay, that is what ma? Female gamete. Here if you see antheridium, this is bearing what beta? Male gamete. This is bearing male gamete. Ascogonium and antheridium will fuse and they will form ascus. Ascogonium and antheridium will fuse and they form what? Ascus. Ascus is diploid. Ascus is diploid. Inside this ascus you have zygote. Inside ascus what is present? Zygote. Inside ascus zygote is present, which is diploid. Zygote is diploid. So by the fusion of ascogonium and antheridium, ascogonium has what? Female gamete, antheridium male gamete. They will fuse and they form ascus. Here ascus is the site of meiosis. Ascus is what ma? Site of meiosis. Inside ascus zygote is present. Inside ascus what is present? Zygote is present. Zygote is diploid and this undergoes zygotic meiosis. What does it undergo? Zygotic meiosis. After zygotic meiosis, they will release haploid spores. Haploid spores. These spores are formed in the sac-like structures called as, these spores are formed in sac-like structures called as, these spores are formed in sac-like structures called as ascocarps. They are called as what ma? Ascocarps. You can see here. Ascocarps are haploid. Ascocarps are haploid, inside which ascus is there, that is diploid. Zygote is diploid. Inside this zygote is undergoing meiosis and it is releasing out spores. Spores are present inside ascocarp. Ascocarp is haploid, do remember, and the spores are also haploid, inside which zygote is there na, this is diploid. Ascus is also diploid. Clear? This is dispersing out spores. After germination, this develops into a new mycelia. Once again, quickly remember, mycelium is having female fruiting body, that is ascogonium, and male, that is antheridium. This is releasing out the female gamete and this is releasing out the male gamete, which forms, all this is happening inside the ascocarp, that is haploid. Okay, and inside which ascus is there. Ascus is formed as a result of fusion. This is diploid. Inside ascus, zygote has formed. Zygote undergoes zygotic meiosis or reductional division, where this is forming or releasing out the ascospores. These ascospores are what? Haploid. Once they fall onto suitable substrata, they gives rise to the mycelium again. Okay, there is nothing to confuse. Everything is happening inside the ascocarp. That is why ascocarploid is haploid, same as fungal body. But after fusion, ascus, zygote are diploid over here. What type of meiosis is happening over here? Not sporic meiosis, that is zygotic meiosis. Next class, if you take, the examples of Ascomycetes include, I will give you a small mnemonic for this also. ACP, nymph, ACP nymph, where Aspergillus, Claviceps, Penicillium, Neurospora, yeast, morels and truffles. Okay, Neurospora, yeast, morels and truffles. ACP nymph, you can remember this. Okay, if you have any better mnemonic than this, you can try for that also, no problem. The third class of fungi include Basidiomycetes, which is commonly called as club fungi. It is commonly called as what, ma? Club fungi. And mostly known forms of this are mushrooms, puffballs or bracket fungi. Do remember all these synonyms of Basidiomycetes asked multiple times. Okay, it is also called as club fungi, puffballs or bracket fungi. These are different types of Basidiomycetes, and mushrooms are also included in which fungi? Basidiomycetes. Morels and truffles under Ascomycetes. Next, Ascomycetes, they grow in the habitat like soil, on logs, on tree stumps and in living bodies and parasites. This is the habitat of Basidiomycetes. Okay. Next, mycelium is septate and branched. What type of mycelium you will see in Basidiomycetes? Of course, dolichopore septa. What type of septa you will see? Dolichopore septa. Dolichopore septa. Yes. Next, if you look into the reproduction of Basidiomycetes, vegetative reproduction is by fragmentation. Asexual reproduction is absent in which class of fungi? Basidiomycetes. Sexual reproduction is through basidiospores. How they are released? They are released exogenously on basidiocarps. They are released exogenously on basidiocarps. Basidiocarps. Next, if you see, the reproduction of Basidiomycetes include, of course, the sexual reproduction of both Ascomycetes and Basidiomycetes include dikaryon stage in it. What do they include, beta? Dikaryon stage, which includes plasmogamy. Dikaryon stage, the sexual reproduction in both of these include 3 steps, that is plasmogamy. Second is karyogamy. Third is meiosis. Once the protoplasmic fusion has happened, that is both the plus strain and minus strain are fused, both the mating types are fused, then there is dikaryon stage that is formed. What is formed, beta? Dikaryon stage, that is N plus N. Dikaryon stage. N number of times asked in the exam, in which classes do you find the dikaryon stage? That is Ascomycetes and Basidiomycetes. After dikaryon stage, they fuse in the basidiocarp. Again here do remember, basidiocarp is haploid. This fusion entire thing is happening inside the basidiocarp. As a result of fusion, basidia is formed. As a result of fusion, what is formed, ma? Basidia. Okay, this basidia is diploid. After karyogamy, basidia which is diploid, inside basidia zygote is present. Inside basidia what is present? Zygote is present. Again zygotic meiosis is taking place over here. What type of meiosis? Zygotic meiosis. Zygotic meiosis happen and this will release 4 nuclei or 4 spores. Okay, 4 nuclei which later turn into 4 basidiospores. How basidiospores are formed? Exogenously. Basidiospores are formed exogenously like this. These basidiospores are released and when they fall into suitable substrata, they grow into again a new mycelium. Ploidy is again important, children. Do remember, this fusion is happening, everything is happening inside the fruiting body, that is basidiocarp. So it is haploid, on which as a result of fusion basidia is formed. After basidia, inside basidia zygote is present. this is also diploid. After zygotic meiosis, basidiospores, those are what? Haploid. Basidiospores are what ma? Haploid. This haploid basidiospores, once they fall on the suitable substratum, they develop into a new mycelium. This is the sexual reproduction that is happening in the basidium. The ploidy, the type of fruiting body that is involved, and the dikaryon stage, these are all the important things to be remembered, imprinted in your mind for the exam. Okay. Next, if you see, fructification is large and conspicuous in which class of fungi? Fructification is large and conspicuous in the Basidiomycetes of fungi. The most Basidiomycetes is the most advanced class of fungi, because here in Basidiomycetes only sexual reproduction happen. Only what happens beta? Sexual reproduction. Asexual reproduction is absent over here. And you can easily remember, there is a small mnemonic which I gave, A pulp, okay. A for agaricus, P Puccinia, U Eustilago, L Lycoperdon, and P polyporus, okay. In this way you can easily remember the class or the examples under the class Basidiomycetes. If you have a better mnemonic than this, you can rely on that also beta. The only point is you need to remember all the examples under this class. All the features which we have discussed just now will be asked by giving the names of these examples only, okay. Next, the last class of fungi are Deuteromycetes. This is called as imperfect fungi because this is called as fungi imperfecti or imperfect fungi because there is no sexual reproduction, that happens in this class of fungi. There is no sexual reproduction. Next, mycelium is septate and branched. Largely they are decomposers and they help. As they are decomposers, what do decomposers do beta? Just now we have learned no, in saprophytes, they will release out the enzymes, they will degrade all the complex organic material and they will release out the simple inorganic materials. So they litter, and those simple inorganic materials, once they are released into the environment, they help in mineral cycling also. This mode of nutrition of Deuteromycetes help in what? Mineral cycling, okay. Asexual reproduction is through conidia again. Now you tell me, when asexual reproduction is through conidia, it is exogenous or endogenous? Exogenous. Exogenous. Next, if you see, when the organisms were placed in the last class of fungi, that is Deuteromycetes, even few fungi which include the sexual reproduction in them were moved out of this class and they are placed again in Ascomycetes and Basidiomycetes. There is a statement in your NCERT, don't get confused. It means that whichever fungi under Deuteromycetes are conducting sexual reproduction, they are placed again under Ascomycetes and Basidiomycetes. The examples of Deuteromycetes, you can remember this by cat, okay. Examples of Deuteromycetes: Coletotrichum, Alternaria and Trichoderma. Coletotrichum, which includes early blight, which causes early blight in Solanaceae members like tomato, brinjal as such, okay. Next, Alternaria will cause red rot in sugarcane, and Trichoderma. All these three examples you have to remember under Deuteromycetes, because whichever characteristics we have discussed under Deuteromycetes, they are asked by these examples only, okay. Now lastly, if we discuss all the comparisons of all the classes of fungi, here we will now fill this table with all the comparisons, okay. Phycomycetes is also known as what beta? Phycomycetes is also known as algal fungi. Phycomycetes also called as algal fungi. Ascomycetes is also called as sac fungi. Ascomycetes is also called as sac fungi. Basidiomycetes, puff balls, bracket fungi, or club fungi. Okay. Deuteromycetes, fungi imperfecti. Next, mycelium is aseptate and branched. In all these three, it is septate and branched. Here also it is septate and branched. Okay. Here what kind of septal pores are present? Pore-like septa. Here it is dolipore septa. What type of septa ma? Dolipore septa. Clear? Next, asexual spores of Phycomycetes include sporangiospores, zoospores and aplanospores. If you take Ascomycetes, asexual spores include conidia. What do they include, beta? Conidia. Here in Basidiomycetes, if you see, again asexual reproduction includes zoospores. Here conidia. Deuteromycetes, again it is conidia. Okay. And in Basidiomycetes there is no asexual reproduction. Here the asexual spores which are formed in Deuteromycetes are what, Nana? Conidia. Sexual spores, if you take, sexual spores of Phycomycetes are oospores for oomycetes, zygospores for zygomycetes. Okay. Ascomycetes, if you see, the sexual spores are what? Ascospores, which are endogenously formed from ascocarps. Basidiomycetes, it is basidiospores, which are exogenously released out of the basidiocarps. Deuteromycetes, there is no sexual reproduction. Examples of Phycomycetes, I told you, right? RAM, mnemonic. Now you fill it. Rhizopus, albugo, mucor. Okay. Ascomycetes, ACP, NYMT, we discussed, right? ACP, it is aspergillus, claviceps, neurospora. Neurospora is mostly used in the genetical work and in laboratories. And unicellular fungi, that is yeast, and edible fungi, morels and truffles. Penicillium is used as antibiotic. Next, Basidiomycetes include agaricus and pulp. A, pulp we said, right? P, Puccinia, Eustylago, Lycoperdon and Polyporus. Next, Deuteromycetes cat, Coletotrichum, agaricus, alternaria, Coletotrichum alternaria, and Trichoderma. Right, if you want, you can take a screenshot of this table wherein the comparative analysis of all the classes of fungi is dealt. So this is the conceptual discussion of each topic that goes on each day. So don't miss out this. This is very much useful for you students. Okay. Thank you.